We work with some of the clinicians, so we get solid tumor samples on a very regular basis where we're freshly isolated from donors. It's very generous by the donor and the family. So we analyze the T-cell responses and just try to understand why they're not working. Not only is the T-cell response we're looking at, it's the microenvironment, what other cells are they, aside from the tumor, that they might be interacting with in the context of that tumor. And that's in the context of an endogenous response. So in future plans with Stephen, what we'll be doing is we'll be characterizing the differentiation of the CAR T cells. And so what my lab has done is we've looked at a lot of very functional responses, and we've defined that at an epigenetic level in human. And so now we can come back and say, this is here's what the cells should look like, right? And here's what the CAR T cells look like now. And the ones that hopefully will be able to correlate an epigenetic definition of differentiation status with persistence of a CAR T cell or the lack thereof, and say this is the program we eventually want to achieve, and then try to either on the front end generate a CAR T cell product obviously with Stephen and others that resemble that, or at least prevent the cells from acquiring these non-functional programs.